All set? Welcome back. We are here to take a look at investigation 2, 3, and 4 from the Diversity of Life. My name is Jean McCabe. I am a retired Boston Public School teacher from the Edwards Middle School, and I'm joined today by
Michael Clinchot at the John D. O'Brien School, 7th and 8th grade science. And I think today I'm here along for the ride.
Okay.
Lots of detailed stuff in investigations 2, 3, and 4. So our student learning outcomes, there are many student learning outcomes for investigations 2, 3, and 4. We're grouping investigations 2, 3, and 4 together because they kind of deal with, once the students have begun exploring what it means to be living, they're now going to explore microscopic life. Mm-hmm. And so investigation 2 is learning the tool that you need to explore microscopic life, the microscope, and then 3 and 4 actually looking at microscopic life. So there are kind of lots of issues. Not issues, but
planning.
Lots of planning
that will be helpful to you. So I have a few things to share with you in hopes it'll make your life a little easier as you go on to investigations 2, 3, and 4.
Excellent.
The first is something I call microscope logistics. Is there a microscope for each student? Do they have to share? Will half the students use microscopes to start with, and others will do an investigation on paper? You need to plan ahead. Are they stored in the classroom? Are they stored on a cart? Who carries them from one place to another? All of those things, because you are dealing with some pretty expensive equipment.
And a new teacher has to find out where they even are in the building.
Ah, yes, where they even are.
And if they have to be shared by more than one teacher as well.
Which is additional planning.
Which is even more planning.
Yes, very true. Okay. So with that said, the first worksheet the student has to do is something called microscope images. It's really important that they take the time and do a thorough job on microscope images, because if they do, then it will make your life a lot easier when you move on to field of view and magnification. The concepts of using the microscope and the lenses, and of course the safety, it comes into play as well. But once you move on to field of view and magnification and you add the a ruler within the field of view so that you're able to use that later on, it is a little bit confusing, and some students are not as adept at using the microscope as others might be.
Patience.
Patience, no question about that.
Because I remember it moves to the left, it moves to the right, it moves opposite, up and down. It's a mirror image, and the kids just really don't have much experience with that.
You're right. And even you can say it 100 times, draw what you see, and they'll still draw that E right side up. And that's not what they see, but they think that's what you're looking for, which is not the case, of course, at all.
Or when you change the magnifications,
yes,
they'll draw the whole E, even though they only see a little bit of it.
This is very true, very true. So this is a baseline activity that hopefully is going to make your life easier as you go along.
Mm-hmm. Yeah, patience.
Cannot say it enough. Things aren't even moving yet in this part of the investigation. Later on, investigation 3, yes, it gets even more challenging. At the end of this investigation, though, is when they get a chance to take a look at the brine shrimp. Now, is anything alive in here? There were brine shrimp, and they were able to, in a perfect world, to see them moving around. So now they've been culturing, and so now they've grown up, they've a little bit larger, and they put them under the microscope, and they can actually see them moving around. It's very cool, very cool. If by chance your brine shrimp are not alive, which does happen at times, there are video clips, of course, at the FOSS website that you can take a look at. They show you about the techniques. They also show you some brine shrimp moving around if the kiddos can't get that on their own.
And this is what you've cultured from the at the end of investigation 1.
That's correct.
Okay.
Provided it all worked out. Yes.
Yeah.
Okay. So the end of investigation 2. Onward. Investigation 3. Microscopic life. Here they are. These are the all-stars of investigation 3. And in a perfect world, when your students prepare their slides, they will be able to see each and every one of these. They'll be able to see movement in those leaves. They'll be able to see the amoeba moving around using its pseudopods.
That's the hardest one, right?
It is the hardest one. The amoeba is really elusive. The others are not so much. With the amoeba, that's a tough one. Oh, the paramecium, of course, everyone's favorite. There's a reading on the paramecium as well. And if you are really careful in your microscope views, you're able to see all of those things going on there. The euglena as well, a little easier, of course, being green, naturally. So investigation 3, let's see, the final part of investigation 3 is the mini pond safari. Again, those are the mini ponds that you started to culture back in investigation 1. And by this point in time, they should be, the population should be flourishing. And you should be able to see quite a few different types of microorganisms. Sometimes you'll have one group in the class whose mini pond is better or more alive than anyone else's, and you can share. The point here is, of course, is that they use the microorganism guide that's in the Diversity of Life resource book, and they're able to either match the microorganisms from their mini ponds or find similarities between the ones in the book and the ones that they found at their mini ponds. Might not be exactly the same, but maybe they move in the same fashion as the ones from the microorganism guide. So it's a very fun way to end investigation 3.
So this is all the microscope based.
Mm-hmm.
So if you don't take the time to make sure that the kids understand how to use the microscope, all of this just becomes even more frustrating.
Right. And I know that some people are tempted to not go through those first few activities, the microscope images and field of view, and they think that they're going to go right ahead to the microorganisms. It's frustrating because the children, I'm so quick to say, I can't find anything. If you've gone through the steps, by the time they get to my mini pond safari,
they'll be able to find
it. Very skilled.
And the other thing I remembered, since we're dealing with middle school students, when you open those mini ponds,
oh yes,
the odor,
they are ripe, no question about it.
So
you do warn them ahead of time. Very
yes.
Good. All right. Very good.
Investigation 4.
Oh, investigation 4. First of all, part 1, investigation 4, the cheek cell scrapings. Due to safety concerns, we just don't create those any longer. Too much body fluids, possibility of hurting themselves, so we pass that. But there's 2 supports within this investigation. One is the cell, the basic unit of life from the resource book, and the other is a multimedia. The kit, the unit recommends that the students work together in pairs or individually if they can, of course, with the ribbon of life, which is perfect if you have the facilities to be able to do that. If you don't, they do tell you how, and it's possible for you to present the ribbon of life as a whole group discussion. Be warned, there is a lot of material, a lot of new vocabulary, parts of the cell, levels of organization, organisms, and then living and alive. There's a lot of information. You do have to do a lot of dancing and singing to keep their attention to present that as a whole group.
I guess it might be a good time to pull out some of those retail vocabulary strategies.
That would be a good thing to do. I think you were right. Good point, Michael. But with that said, I mean, it can be done certainly, but there is a lot of, just know that there's a lot of information. Okay.
And then therefore maybe a lot of information that a teacher needs to become refamiliarized with.
One thing I would say is do not go to the ribbon of life without having gone there first.
And I think that brings us to the end
of our 234.
Of 234. So hopefully they found that helpful.
Hopefully.
And we'll see them back again. We'll be back again for 5, 6, 7.
5, 6, 7.
5, 6, 7. See you then.